It's my great pleasure to be here and to speak to you about a very similar topic. So we are kind of going through similar concepts which Luke presented in my talk and looking at them and discussing them, hopefully, with you. So this is an example of classical pleiotropy, which we usually would know about if we did medical school in 90s or something like that. I didn't do medical school. So albino individuals lack pigment in their skin and hair. They have cross ties at a higher frequency than pigmented individuals. And genes that causes albinism can also cause defects in the nerve connections between the eyes and the brain. These two traits are not always linked. So first of all, we discussed common and rare diseases. So please raise your hands if you think this is a common trait, albinism. Do you think this is a rare phenotype? Please raise your hand. Let's do some physical. Wow, chairs agree with me. Sorry about that. Yeah, so this is a rare trait. And what you have taken forward from the Luke's presentation, do you think, and now think about two types of pleiotropy, where there is mediation, so this is a vertical pleiotropy, or it is a horizontal pleiotropy. So raise your hand if you think this is a horizontal pleiotropy. Do you think this is a horizontal pleiotropy? Yeah, don't hesitate. I think, yeah. So here, one gene causes two phenotypes, which are not necessarily occurring in the same individuals, and they probably don't cause each other. So most likely, this is actually horizontal pleiotropy. And thanks for your support. So I will show you some examples today with some methodologies, and we'll discuss multi-phenotype approaches. So pleiotropy is about multi-phenotype effects, and let's start by thinking about multi-phenotype effects on examples, right? So what we usually do in GWAS, and probably most of you have done some kind of genome-wide association study analysis, we run individual phenotypes, and this has been happening since 2008. We were lucky to have body mass index measured in almost everyone in our studies and blood lipids. But this causes a problem, because if we observe the same association at different loci, at the same loci for different phenotypes, it means that we need to understand what is actually going on at very specific locus, right? So here you see an example from 4-lipid GWAS, and you can see that there is a huge overlap between these four phenotypes. And Luke has already mentioned at top level that they are related, they are tightly related. What you could notice, and probably would suspect, that FTO locus somehow is associated with lipids. And in this example, it is associated with both triglycerides and HDL cholesterol. But even more strikingly, in the middle, right in the middle, there is fatty acid desaturase 1,2,3, a locus which is associated with all four lipids. And there are other examples which you probably know better than me. But I'm just highlighting these two for you to make a different example. Now, let's look wider at metabolic traits and how they share associated loci. So here, loci, as in the previous slide, are named by the closest gene, right? So we haven't run colocalization, which Luke mentioned before, but just by name and because of the correlation between these phenotypes, we think that there is a lot of shared genetic component. And what you can find on this plot is that FADS1 is also associated with glycemic traits, for example, with fasting glucose, as well as with lipids. And next, if we look at FTO locus, it is expectedly associated also with glycemic traits, obesity, lipids, and also we have here type 2 diabetes represented. Since this plot was done almost 10 years ago, probably it is actually associated with all mentioned traits on this slide at the current state. Now, I want to make you another example of using LD score regression, right? So we discussed that there are many phenotypes and we can explore how they are correlated, and we can find some logical correlations, so possibly global pleiotropic effects, if we wanted to call them like that. So when we analyzed random glucose as a phenotype, we decided to explore how it is related to a variety of other phenotypes. So we used LD score regression database, and I think there were more than 400 phenotypes. So here, there are only those which were at least nominally associated. And usual suspects would immediately jump into your eyes, right? So a pink triangle highlights to you all obesity phenotypes which are positively, directly correlated with glucose levels. And the blue rectangle shows to you glycemic phenotypes, right? They are all correlated. Luke also mentioned that HbA1c is correlated with levels of glucose in the blood. We have fasting insulin. We have type 2 diabetes, which develops after a long time of having high glucose levels. So it is logical. Only one phenotype, which you probably are asking me about, this is HOMA-B. This is a phenotype indicating reduction of beta cell function in pancreas, because when people have diabetes, their pancreas is suffering pretty much. That's why it is logically inversely correlated. And on this picture, there is an interesting set of phenotypes, bright green and dark green or turquoise, if you want, where we see that some phenotypes related to lung function or lung affecting lung are also either directly correlated, such as lung cancer or squamous lung cancer, as well as forced vital capacity, FVC, and FEV1, forced expiratory volume. They are common traits measured in many people, and they are quantitative. They are inversely related. So pretty much when potentially when we explore this relationship further, if we wanted to ask a question about causality, we have some indication about the fact that potentially if people have higher level of glucose, they might have reduction in lung function, and instead lung cancer as a disease might also be related because there might be some shared risk. So I'm not going into Mendelian randomization of this, but we indeed have seen inverse effect in the follow-up analysis through MR between random glucose and lung function traits. But now, let's go back to the global picture of what we have when we look at the associations with individual variants and at individual loci, right? So you will be probably kind of struck by the number of associations reported for some specific loci. And they are usual suspects, which we knew about many years ago. So the most associated locus is ABO. At some point, I checked GWAS catalog. It was 1,245 unique phenotypes. FTO, I think this is not updated information, 90 unique phenotypes, right? And ApoE, you also know what this where this locus is associated, the phenotype effects. It was associated with about 60 traits. And then you can think about HLA loci, where we need to dissect very precisely what is going on, but associations are striking at many loci in human genome. We have many phenotypes affected. Right, and going into how we started to think about these multi-phenotype effects at individual loci, we could look at the very early report in Diabetes in 2008 by Friese et al. About the effects of FTO on a variety of quantitative metabolic traits, right? So you can pretty much draw a line, diagonal of the effects and associations, right? So many of these phenotypes we have shown subsequently are causally affected by obesity or adiposity, BMI, right? So we have seen this association, and we have done further analysis later on to dissect the effect. So pretty much here, we will be speaking about the vertical pleiotropy, right? That's the effect which we are exploring in Mendelian randomization further. Now, we have many other levels of omic data. And if we think about metabolome or proteome phenotypes, they are highly correlated. And that's why the previous speaker mentioned about the fact whether we are actually in a need to start dissecting the relationships between them, because they are so tightly related that they might be just different steps in the metabolic, in the biological processes. And here you can see similarly, many metabolites, mostly they are lipid metabolites and related to fatty acids. You can see that there is very high correlation, either inverse or direct. So when we try to analyze them potentially together, we need to decide what is to be analyzed to avoid collinearity issues and other problems with statistical analysis. When we think about multi-phenotype analysis, people started developing methods almost a century ago, and we always think about different ways of evaluating these multi-phenotype effects or pleiotropic effects, if you want to. So we might want to reduce the dimensionality of our data, like from the previous example where we had many metabolites, we want to select some of them. We want to develop multivariate models. There are some graphical approaches and many other. So in the 90s, we started to work with linkage analysis, and it was successful for rare phenotypes. For common phenotypes, it was largely unsuccessful. And recently, we have been lucky to have an ability to analyze a variety of common phenotypes and focus on common variants. Now, when we start thinking about multi-phenotype analysis, we can analyze phenotypes together. So we will identify biological advantages, so pleiotropic effects, and we might have statistical advantages by analyzing multiple traits together, so taking power, improving our power from combining information from multiple phenotypes which are correlated, for example. And we can improve the precision precision of parameter estimates in this analysis. So quite a long time ago already, this paper by Gail Lute highlighted a set of types of analysis which can be used to combine multiple phenotypes in one genome-wide association study, so to conduct multivariate GWAS. And as you can see here, you can subdivide them into main groups, those which are direct methods, indirect methods, and actually use univariate analysis. So univariate means single phenotype analysis. And this is the top right corner is the analysis where we analyze each genetic variant for every single phenotype or trait we want to explore. But then there are also different ways which combine these genetic effects in meta-analysis by incorporating correlation between phenotypes, and we might run in issues which were highlighted by Luke, because we have genome-wide correlation, but if we do a point analysis, single variant analysis, this global correlation might not be exactly the same as in the single locus. And there are other meta-analysis methods, but also you remember methodology which reduces the number of parameters. So, for example, there are some approaches which propose to calculate either principal components or factors and then analyze smaller number of components or factors against DNA variation. And there are also direct methods. So one of those direct methods is one which I quite like, because I'm focusing on this methodology directly. It was first proposed in the Multifen software, and this method uses reverse regression approach. So you can see that instead of analyzing genetic variant, we are actually predicting genetic variant from the traits. So what we do in multivariate GWAS when we want to dissect these multi-phenotype or pleiotropic effects, first of all, we try to gain power as compared to univariate analysis. And actually, this paper has conducted some analysis and made inference that use of multivariate GWAS methods can be recommended even when genetic correlations between traits are weak, but still some correlation exists. And many examples which we are making today are making use of large biobanks which feature a variety of phenotypes measured for many individuals at the same time or for the same individual at different time points. And this is actually brilliant. It enables a lot of exploration of phenotypic variability, and we can also address potentially still possibly existing missing heritability question. Right, so how we are doing the reverse regression approach implemented initially in Multifen. In reverse regression, we use genotype as outcome, and phenotypes become predictors. This sounds weird, but mathematically it is correct. So in traditional GWAS, we would have actually the same phenotype evaluated for each SNP, right? But we can run reverse regression where we introduce the model where for each SNP we evaluate multiple phenotypes at the same time. So that's why it is called reverse. We implemented it together with Andrew Morris and Radik Magi in the SCOPA software tool for common variants. And why I like this software, it's because we can use it for multiple phenotypes, and we are working now on implementing it for UK Biobank file format. And then we can actually, if we analyze 4 phenotypes, we can evaluate full model with 4 phenotypes, select the variants which are associated, and then look into the individual loci to dissect what is going on at this specific locus at every single time. And if we do the same modeling of phenotypes, we can also meta-analyze different data sets in one analysis. You can also run the same approach for rare variants. Obviously, it's this one is not, this analysis is not focused on individual variants. It is gene-based or locus-based, if you want to. And within the rare variant for reverse regression, we are counting the alternative rare alleles within the locus and calculating the proportion of these alleles as outcome. And you can see this counting, right? So how many variants bear rare alternative allele 1, 2, and 3. That's why we are making a proportion of 3 out of 10. And that's why we can calculate all other analyses similarly to common variants. So it is a collapsing method. Now, I want to show you one example where we analyzed 31 NMR metabolites. So we have used the correlation between metabolites to exclude some of those which are tightly correlated, and we're left with 31 out of approximately 200 something, right? And the usual suspect of FADS1 locus was showed the strongest association. So one interesting point here is that if we also, and I will highlight it later, we actually compared our ability to run analysis of these many phenotypes by using complete case scenario. What it means that standard software tools, when you run them, they will exclude any individual which doesn't have a phenotype, at least one phenotype measurement across many, right? So if I use complete case analysis, I only have 4,000 people. If I'm able to impute using correlation structure, then suddenly I have much larger data set and power goes up. So here you can see the associations, and this is a full model analysis, so pretty much 31 NMR metabolites. But if we go into the detail, and here I left only standard blood lipids. It's another example for FADS for FADS locus. So here I left in the graphs BMI and 3 blood lipids to show you the results of this analysis. So as you can see, FADS is usually very significant when we combine effects from multiple lipids. But when we start comparing the effects which we calculated in this modeling exercise, so the software is able to run all models which we can create with 4 phenotypes, BMI and 3 lipids. And you can see that the association is observed with triglycerides, which is where beta is negative, and with LDL cholesterol, where beta is positive. Interestingly, it seems that BMI is not affected, is not affected at this locus, and HDL-C also is not. So what we see here is that despite looking at the initial association, which I show you with all 4 lipids, we do see LDL-C and triglycerides. Total cholesterol is not present here because it is almost 100% correlated with LDL-C, right? So we don't analyze it in these multi-phenotype models. If we look at the other example, which I highlighted to you before, and you know about very well, so here BMI is shown with this purple diamond. You can see that association with BMI is very strong, and the specific allele we were looking at is providing us with negative beta. But the p-value of the overall model is actually slightly less significant as compared to the single phenotype model with BMI. So that tells me that the primary driver of association in this locus is actually BMI, and effects on HDL-C and triglycerides are really tiny, so possibly it's a secondary effect, or basically they're minimal, and the secondary effect which we see is due to vertical pleiotropy. So these are multi-phenotype methods which we are proposing for GWAS. We can also run the same approach with methylation data and other data types if we want to. So methylation data is becoming widely available. We have multiple points in the genome, and you know how it is working. There is an addition of methyl group, which we are looking at, specific points in the genome. So if we apply the same modeling and invent multi-phenotype EWAS, it is actually even possibly more plausible biologically, where we have methylation data as on the left of the equation, and our phenotype at individuals is on the right hand of equation, because in EWAS sometimes we discuss these effects in both directions. So we implemented a similar tool in Methylscopa, and just to show you an example, when we analyzed the same data sets, Northern Finland birth cohorts, as in previous examples mostly used, we have seen an association at EWAS significance level at a locus when we analyzed fasting glucose and fasting insulin. And if we were analyzing them separately, we wouldn't see this effect. So there is correlation between glucose and insulin, and this approach has used the correlation between phenotypes to enhance power. Right? And one final comment on multi-phenotype analysis. When we start thinking about GWAS consortia, there are many of them. They analyze data in a single phenotype analysis. Individual studies which have many phenotypes contribute to many of these consortia. So pretty much on right-hand side, you can see studies which contributed to all those consortia on the left. And we already said, correlated phenotypes. So as we mentioned, we can run PheWAS approach, which looks at individual variant across the whole range of phenotypes. You can see that in LocusZoom online tool, where UK Biobank data is provided also for every single variant they can find in the data. But we can also do multi-phenotype GWAS. And I already mentioned to you that if we have missing data in the phenotype, it's an issue. We are limited to the complete case studies. So if we do this example of having 5 phenotypes in single phenotype GWAS, and let's say 5 individuals, gray squares here are missing data points. So if you run complete case GWAS for single phenotype analysis, you have nothing else to do. You have 60%, 100%, 80% data available in each individual study. But if you start looking at the multi-phenotype analysis for these 5 phenotypes, which are in columns, across individuals horizontally, you will have data only for 20% of cases. That limits our power. And then we need to deal with missing data in omics, right? So we want to study pleiotropy, and that also possibly discusses the point from someone who was asking question, what do we do when we have different power? Obviously, we need to try to improve power of our studies, and here, when we do complete case analysis, we lose power. In genomic data, somehow, brilliantly, from the very beginning of GWAS, we introduced imputation. So when we run multi-omic data analysis or multi-phenotype analysis, we need to think how we can impute phenotypes. And there are some approaches proposed for example, for summary statistics imputation. It is, again, genetic variant imputation, but we can impute phenotypes, and we can gain power in biological data sets and biological insights. So thinking about imputation approaches and going into mentioning some machine learning approach, we have standard approaches. We mentioned complete cases. We can do mean imputation. This is merely just inserting the same information for everyone, so we are not improving our analysis, right? We can have statistical approaches, single imputation or multiple imputation, and we can also start looking into the machine learning approaches, which are potentially very useful for one type of phenotypes or another, and we can explore many of them to to decide what is the best. So my PhD student worked on these approaches, and she evaluated how the approaches perform. She selected here, I think, 6 different approaches. These approaches, every one of them addresses different missingness data mechanism. Just to introduce a new term for you. So there is missing completely at random, not at random, and so missing at random, missing completely, and missing not at random. So you see that some approaches are fast computationally, but when we deal with hundreds of thousands of individuals or phenotypes for them, it's important factor. And we need to think about the availability of data which can be binary, ordinal, or quantitative, right? And the need for a statistician or analyst to do parameters tuning. When we need to do that, it is another potentially subjective introduction of a subjective step in the analysis. So when there is no need for parameter tuning, it's better. So we decided that random forest was doing the best for imputation of multiple phenotypes, and then she explored variety of options in R and how these options can work with high-dimensional data, or whether they can work in C++. So the solution was to do the random forest-based software in C++. And here I'm showing to you the same example, right? So there is example with 4,578 people and complete. So this is complete case analysis. This is imputed. In a very small data set with 31 metabolites, we improve power by adding 2 loci to the analysis, and this helps us to improve power of our locus discovery. So there are some publications or bioarchive methods for this, and I think phenotype imputation is coming into play to reduce the differences between availability of phenotypes and how missing data is generated sometimes with specific technologies. Now, another point which we sometimes want to discuss, and I will just focus on it very briefly, it's the longitudinal look at the disease development, how we can try to look into relationships between phenotypes. So just very briefly, in the ECC consortium, we love to look into the birth weight as a Here you can see that there is a group of loci which are actually associated with adult height, right? So this is logical. Birth weight is in some way also a reflection of height. It's very difficult to measure height of the newborn baby. And there is also a group of phenotypes which are mainly metabolic phenotypes, and the loci associated with birth weight have inverse effect on these traits. So pretty much we say the higher the birth weight, the lower the risk, or we need to turn it around. Lower birth weight is associated with higher risk of potentially multiple metabolic outcomes in adulthood. So this is another effect which we might observe longitudinally. But there is a trick. And this effect is easily observed for some loci which are also associated with diabetes. This is a fetal insulin hypothesis, which says that if insulin is lower in the baby, produced by baby, then we have a higher risk of diabetes. So pretty much if birth weight is lower and diabetes is higher. But if we go into the loci which actually affect maternal genotype, so mom produces more insulin, it is diabetic uterus effect, then these loci are directly correlated with the risk of metabolic conditions in adulthood. So you see, this also links to the effects which were discussed before, right? So we have global correlation, but at local level, we need to look into the biology of the effects of each gene, of the biological effects within loci to understand the potential relationship between phenotypes and biology. And so basically, if you want to run Mendelian randomization, think what you are actually putting together, right? So one last comment about longitudinal data. So longitudinal data is very expensive. You pretty much start looking at for moms which are about to give birth and try to follow individuals for decades. And you stay in contact with them. So there are excellent examples of such studies. I was giving to you Northern Finland birth cohorts, ALSPAC study, and some other. This is an excellent kind of opportunity to explore how longitudinally the phenotypes change and how people develop conditions. Another study is Whitehall. So here, you need to look at this graph. It is very interesting. You need to look from the right to the left. So 0 is the year when people develop diabetes. And there are 4 measures, continuous measures of glycemic phenotypes, which are related to the risk of diabetes. And you can notice that basically it is very important for us to notice when there is this change of and when the liability model can be applied, right? So there is a quantitative phenotype that changes in time, and then it leads to a disease, right? So when we look at the fasting glucose at 0, when individuals develop diabetes, the change is visible pretty much only a year before the onset of the disease. And you can say to me, possibly it might be easier to look, for example, at 2-hour postprandial glucose. It's a test, so people need to come to the clinician and drink a glass of sugar drink, right? So this is a very expensive test. There are some other measures of beta cell function, which, as I said to you, in pancreas, the organ or beta cells start to compensate issues with insulin resistance, and then they fail, right? So longitudinally, the processes and what we are able to detect is changing differently for different phenotypes and different point of times, right? So when we start modeling these effects for some specific phenotype and we want to explore what is going on with related traits, right? So we can evaluate variety of factors, right? And we can say, oh, there will be some pleiotropy here. So we can have classical risk factors, biochemical data, genomic data, metabolomic, epigenomic data. And
2 minutes more.
Yeah, okay, finishing. So we can look at the disease onset, binary phenotype, and we can look at the quantitative phenotypes, right? We can run association studies or do prediction. So with association studies, we are good. We have done a lot of them. With prediction studies, we can explore a lot by using variety of methods and variety of data sets. So pretty much in this exercise, we explored epidemiological data, biochemical and metabolomic data, and epigenetic data. So when we combined all this data in Northern Finland birth cohort, we actually were left with 513 individuals. At that time, we decided not to do imputation. And then we explored how we can play with the phenotypes, whether we need to transform them or we can combine them as a raw or as scores and exclude them. We did some modeling, and in the end, the best prediction for phenotype, so we had only 2 time points, and the best prediction was done for fasting insulin. And you will say to me, okay, the improvement is not so big because classical prediction gives us a lot. Then there is metabolic phenotype, there are metabolic traits and epigenetic data. But still, we are able to improve the prediction in time point 1, right? And to do that, it is actually useful to combine information from multiple omics data levels, explore relationships between these data, and possibly explore pleiotropy. So overall, big omic data is becoming a reality in genetics. So I invite you to think about pleiotropy, implement the approaches which you like to combine information from multiple phenotypes, and think about missing data when you work with such multiple phenotypes, as well as trying to implement new ways of analyzing data, maybe using machine learning and AI. And thank you for your attention.
Please come to the mic if you need to ask any question.
Thanks for the great talk. Could you comment on some of the methods that use summary statistic data, for example, multi-thread analysis of GWAS or MTAG, and also for individual level data? I think there are also another family of methods that basically use PCA. So you run PCA to reduce the dimensionality of the multi-phenotypes and then run GWAS on those PCA. Could you comment the difference between that methods?
Thank you. This is an important area of exploration, because to me this seems that we have opportunity to, there are some methods which start appearing. I don't know specifically for summary statistics. So obviously, if you can develop this method or you find this method, it offers you combination from summary statistics, right? So MTAG, possibly, right? They have some pitfalls which might be related to issues which have been mentioned. And you said another one?
I think in individual level data, you basically just run a PCA on the multi-threads.
Yes,
and then
so
run
for
GWAS
PCA analysis, it's about how you interpret those PCs. So you need to understand what this PC is telling to you. And sometimes it's really difficult to dissect afterwards once you have these PCs measured. What do they mean at a specific locus?
And what about the methods that like SCOPA, for example. So it's like one
So
single trait. So like, how do you interpret that?
Basically, we are in this SCOPA. You can model the full model and then run each possible model of combination of phenotypes, including single phenotype model. And then you compare all this set of models using Bayesian information criterion. So you can select what is the best combination of phenotypes at this specific locus.
Okay. Thank you.
Excellent talk. Is it working? Or yeah. My name is Yoon Sung Lee from Norwegian Institute of Public Health. It's regarding. It's all about. I have a question about the multivariate GWAS. It's a great idea. So outcome variable is the allele, the number of alleles at a specific locus, and the X variables are like multiple different phenotypes. So is it like Poisson regression? Did you run Poisson regression, or did you just like run an ordinary regression, or
to do what?
The when you do the multivariate GWAS, X variable is number, like it could be like 0,
do you
1,
mean
2.
to evaluate how phenotypes are related to each other?
Yeah.
Of course. We need to know that we don't generate collinearity issues. That's the point when the phenotypes are almost the same. We cannot distinguish between them. So we need to know the relationships between phenotypes before we select them for multivariate analysis, if we run multi-phenotype GWAS.
And did you include PCs to address population stratification?
You can create residuals and include it in the analysis, of course.
All right,
It allows you to do everything if you decide to use this approach.
Okay, fantastic. Thanks.
Hi, Linda Pulfis from Ambry Genetics. I had a question about the methyl SCOPA. In that model, you showed the summation of the phenotypes. Was that based on prior information, or did you just include all the phenotypes as a summation?
For methyl SCOPA, again, we run the exploratory analysis on two phenotypes, which are, yes, correlated, fasting glucose and fasting insulin, but they're not as tightly correlated to generate issues with collinearity.
Okay. And then I had another question. Some of these traits are on the disease continuum and are biomarkers in blood, and then are a disease outcome. So is there any work focusing on the expression level in the tissue of interest for the disease outcome?
Of course. That's back to the previous speaker. So you absolutely need to do colocalization to understand what is the tissue involved, what tissue is involved, where this gene is expressed, and you can run TWAS, etc. So basically, we obviously need to do all further steps to understand biology better.
Thank you.
I think this question concludes this session. So thank you very much for everyone for attending.